members. We had a meeting today with LUFA, and it's not looking very good. The administration is not budging. They just want us to say, cut our salaries by much. But as I was telling to the TAs, that is like adding a lot of rice in a bag that has a hole underneath. We went through this 8 years ago. We had a freeze in salaries for 2 years. And here we go again. And we say we can freeze to roll out for next year until we get out of this COVID thing and we renegotiate next year. They don't want to hear anything about it. We have asked for showing us the financial papers that demonstrate that really we are in a hole and that we absolutely, if we don't do anything, we will be in jeopardy. And they don't want us to show us anything. So they are supposed to show us something today, and they haven't yet. Today is the last day, otherwise we're gonna go to the Board of Work and file a complaint for bad faith negotiation.
I'm so sorry you have to deal with this. Yeah. With all the stress, the semester starting and homework and probably personal stuff, you also have to deal with
that. Yeah.
I can swear in Russian.
and you know, and it's like I'm being 15 years, I had a good salary, but I'm the only income at home. So if I need to cut my salary, well, that's not good, right? And there are younger faculty that has a way lower salary than mine, and we have sessionals and we have master lecturers for which their salary is not good. So if we ask them to cut also their salary, well, they're gonna be behind for retirement. We were doing the calculations today. If we give somebody has a salary of $100,000 from now 25 years, that means about $257,000 that they don't gonna have. That's a lot of money. Okay. And because it's not just that we're gonna take a back in the salary, is that we're never gonna get back to where we should have been. So I just wanted to transmit that information to you guys so it doesn't come as a surprise that because sometimes these mandates of a strike comes very quickly. Sometimes it's overnight. We have been trying to negotiate since April last year, and nothing has come through. So I just wanted to let you know, so if it ever comes to a surprise, we are ready to strike. Not to affect you, because we do care about you, but the university really wants to cut a lot of positions. There are 75 positions since last year that were lost through retirements and didn't get replaced, and they want to cut more programs. Last summer and fall, you heard about the closing of many programs, right? And they want to close more programs, which means they're gonna cut more faculty. So we do this because we like to teach, we love our jobs, but we also require good conditions. The library is pitiful. You cannot find new journals. We have subscription to journals, and now we don't have, if you know the amount of money that the library gets for function, is laughable. So when you try to get journals through the library and it's a block, that's one of the reasons. So we cannot promote higher education and a good quality program and pretend that we are a first-class university when the resources are not there. So this has nothing to do that we don't care about our job, about your future, but conditions gotta be. See, Dominic Giro, and you can say it, that I say it, he did a mess at this institution. He destroyed a lot of the economy by going through a very campus.
So I find that
was a whole
has been messing up for good decades, probably at least 10 years at this point. I've been hearing about my older peers that Laurentian tried to make them pay twice for their semesters, that they were bad for paying out not reimbursement, but the scholarships.
Personally, no, they take the money very quickly, but don't give it back very quickly.
Personally, I was affected this year. In around April, they fucked up in the list for being an designated learning institution, which is a requirement to get a visa to come to Canada. I'm an international student. I pay 2-3 times more than any other Canadian student, yet they allow themselves to do that to us. I don't know what about your guys and which programs you are, but my annual tuition is $25,000, plus minus, give or take. And
plus cost of living and etc. Yeah, it's high. I was an international student for many years too, so I know where you are exactly right now, Elena.
Yeah, and seeing Laurentian just doing all of that and trying to get away with it, and their programs are outdated. Professors, well, a lot of them aren't keeping up to date with the way the course should be and missing a lot of points. So
yeah, well, that is unfortunately something that is called academic freedom. That we try to fight sometimes, and Amanda knows about it. How when we complain about it, and Amanda knows I've been very advocated to ask faculty members to upgrade their courses, and so, well, you have no say in my course, Mary. So anyways, I just wanted to have that parenthesis and tell you this because you are my primary concern. Okay. If you weren't here 3 years ago, when we go into lockdown, it's a lockdown, which means that they cancel. We don't have access to our emails. We don't have access to D12. You cannot contact me unless you have my phone number. And very few have my phone number. So, Anish, you have my phone number. And Emma has my phone number. But other than that, we cannot contact you. So normally we go around a little bit. The TAs normally are in contact with me, but they don't have access to what I have. Okay. So when assignments are given, if we have a due date for them, like we have a due date for the video, you submit your video and things like that. So there are some rules, but normally they just immediately, once we call for a strike, they cut us off completely. So that's it. We'll see. We're going to continue negotiating and we keep you posted.
Mari, you can go first.
Yeah, I want to just add something with all due respect to the committee of the board or whatever responsible for the university. But have you heard a Black comedian named George Carlin? I guess most of us know him.
Mm-hmm.
He spoke about something like that in order to cut funds and all that. This is because it's just a business. The outdated or the backdated curriculums is just because I think there is something to do with politics here. But seeing all this, they are cutting funds from people whom they should add more funds because they are raising new generations of people, where in fact the CEOs of companies who are just keeping track of their money, they are getting paid $1,800 an hour. So I find this very, very pissing. Like, what is he going to do in order to just be the CEO of a company where in fact the people who teaches us new things and good things, because actually they are enlightening our brains, they are getting cut off. But people who are just getting or improving their financial income are the ones who are getting paid higher. So I find this very irritating.
It's costly. We receive some support from the province, but it's not enough necessarily to run a university like ours. Of course, attrition is something that can help us to get out of the hole, but a lot of students prefer to go to a bigger university. You came here for diverse reasons, right? I like the fact that we have small classes, like this one is 60, seems huge, but this same class in another university can be 200. And I get to know you. Those that have taken courses with me, you know that I'm very personal with my students in class and in labs. So that's a different dynamics that we have here, and that's one of the reasons why I like Laurentian. But at the same time, we need to run. We need to run these labs, and we need to run. And we have been cut. This department in biology, if you're in biology, we used to be big. We were still like 21 people. Now we are 16 faculty. Okay. And Sarah can say, yesterday we were in a meeting. And we are looking into how we can deliver our program in biology with the less impact on you guys. So we're going to have another retirement. It doesn't going to be replaced until the next 3-4 years at least. So how we can program the courses for you to have still a good experience in the department and complete your program. And that meeting was for 2 hours almost yesterday. So we are working hard into trying to not affect you. And then we need to deal with the administration wanting us to keep cutting. And the new dean in science and engineering needs to cut budgets. Again, so it's hard, but I don't want to extend more on this, but I just wanted to keep you posted, okay? So we're still in negotiation, and this is the reason why my background says what it says. I tweeted it, they want us to advertise it long, and I think there are a couple of them. It's in my Facebook, it's in my Instagram, it's in my Twitter, so the more we spread the word that we support higher education and here at Laurentian with the less impact, that will help to pressure also the administration. That's all. You don't have to do it if you don't feel that your heart is on it, right? Because I will never force anybody to do anything that you don't want to. Okay? Okay. Yes.
Would you be interested in, if everything escalates to the point of the strike, will you be interested in keeping in touch with the students?
I shouldn't.
Okay.
Not that I don't want to, because Amanda went with me through last negotiation, and she knows that I was passing messages to my students through her. But you never heard this from me. I have never said this.
Oh, yeah, obviously. Just in case you never want us to contact you, Telegram might be a very good option for secure chatting and exchanging information for those who don't want to talk to each other.
Perfect. I will take into account that, Elena. Okay. But the TAs will be able to keep contact with you, and through them, I can not passing any messages to you. Okay. But I will always deny that I have contact with you. Otherwise, I will be in trouble. Okay. So, article discussion. We had 2. Kaya, even if you send me an email, I won't be able to read it because they block us completely. We don't have access. They just turn off the switch for D2L and anything, any platform of the university. Remember, university owns our email accounts. We don't own our accounts. The university owns our accounts. We are not the owners. So I could even look at any of my emails.
I have a question. Is this for just the biology department or the whole school?
The whole school.
If they go on strike, it's the entire school.
It's the entire school.
like 4 years ago. So
Yeah, exactly.
I was taking nursing in college back then. So second strike.
Yeah, and if we go on a strike, come and visit us.
Then do you mind writing your address so I can maybe bring your chocolates and muffins?
Normally, I always ask to be in the School of Architecture.
Okay.
Because it's close from home. So that's normally where I go. But Amanda, just through Amanda, you can get a hold of her. Amanda will let you know where we will be. Amanda Durkin is, I should start with the introduction of my wonderful TAs. I'm so sorry. Not neglecting. So here we go. Amanda.
Hello, everyone. I'm Amanda. I'm a PhD candidate in the Biomolecular Sciences program, and I'm doing immunology type research at Health Sciences North Research Institute. And I've been a TA for Mary for many, many years. Done Devo quite a few times. It's a great class. And yes, if we do go into a strike, you guys can email me and I will come up with answers.
Sarah?
Yeah, hey guys. So I'm Sarah. I'm in my second year of my master's right now. I work with Dr. Albert Schultz-Hosteda. So my research has nothing to do with developmental biology. I do research on Vancouver Island marmots, which are like big kind of groundhog things. So yeah, I did Dev Bio in my undergrad and I really liked the course. So I'm excited to do it, but it's going to be a bit of review for myself as well. But yeah, I mean, if there's a strike, I will do everything that I can to help you guys out for sure.
Then we have Jenna.
Hey everyone, I'm Jenna. Like Sarah, I don't necessarily have a background or I'm doing my master's in developmental biology, but I am just in my first semester of my master's in Dr. Jackie Litzkus' lab. And my project is looking at road mortality mitigation strategies for vulnerable turtle populations in Eastern Georgian Bay. My undergraduate degree was in environmental science at Trent University, so I took a lot of biology courses there. And yeah, I'm happy to be the TA for this course. Excited to see all your work and learn a little bit more as, we go. That's me.
Thank you.
Jenna.
So Sarah has TA for me in the past, but for vertebrate physiology. Amanda, she is my go-to girl for this course. This is the 5th time, Amanda, that you take this course? Or is it 4th?
I think it's 4th.
Yeah. So, and that's because Mommy is not with us anymore. Mariam, I think she TA'd this course like for 6 years. Even when she was an undergrad, she TA'd it for me. So, and last but not least, we have our technologist, Joe Felix.
Hi, everyone. So I'm Joe. I actually graduated almost 2 years, 1.5 years ago with my PhD in boreal ecology. And well, I did some postdoc work in between and couldn't find anything end of last year. So I got hired as part-time technologist with reduced hours too now. So yeah, I feel that they don't have enough money. I mean, they should have the money, but like, yeah. And I basically only prepare the materials for Mary.
Jenna.
And yeah, and I worked on wildlife diseases in the Northwest Territories.
Lena, you had a question? Yeah. Amanda, Sarah, and Joe Felix, and Jenna, could you please add your email to the chat, please? I was about to ask that. Okay, so we're going to start the article discussion. Today was a little longer just because I wanted to do all these introductions. But we normally do the article discussions by article, okay? And the discussion, you guys do the discussion. We guide and we can answer some of the questions, but you guys are the ones that do the discussion. If nobody talks, it won't be any discussion, okay? So we're gonna start with the paper that I think most of you read because it was the shortest, not because you were more likely to read that one. And that is the Planaria paper. So, Emma, would you want to start, please? Just to break the ice.
Yeah, absolutely, for sure. I'll definitely start. Okay, so I found a couple of really interesting things about the Planarian Regeneration article, and some great discussions too in the group that I had, which is great. The one thing that really stood out to me was that the stem cells that Planarians use for regenerations from their segments, the Neoblasts, I found it very interesting that they can only grow and divide in Planarians. So I thought that was pretty interesting to learn about, because I don't know a whole lot about Planarian regeneration. So I thought that was a pretty interesting piece of the article.
Okay. Anybody else wants to complement the information that Emma just, I know that a lot of comments were in that paper because I download them all. So I know exactly who asked a question, who made a comment, who replied to a comment. I have all these stats. So I know that a lot of people read that one. Not all at the same time. It's a huge cacophony. Go, Elena.
If a strike happens, are you going to be having access to a parasol?
You guys, yes. That's outside the university.
And you?
I will have access to parasol. That's outside campus.
But that's outside of the discussion. Okay.
Sorry?
That's, I assume, outside of the discussion. Like studying is not happening.
Yeah.
Okay.
Emma mentioned her group where yes, you are, Kaya, you are assigned. You don't know necessarily who is in your group unless you interact. I know who was in each group, and each paper going to have a different grouping. So you were in groups because I cannot have one group of 60. That's too much people to interact. Who else wants to speak about that paper? So Emma was talking about those special cells that allow regeneration. What type of cells are those cells?
Neoblasts.
Neoblasts.
And then blasting.
Okay. So those are neoblasts. And what is one of the specific traits of that neoblast?
Specific trait of neoblast?
Mm-hmm. What made them special?
The proliferative.
Robert?
Yeah, the pluripotent, the neoblasts
Are they totipotent, pluripotent?
I believe they're pluripotent.
Okay, and what is the definition of a pluripotent cell?
That they can regenerate or proliferate and differentiate into multiple different cells. Not all, but a large.
Perfect. That's really good, Robert. So they cannot differentiate in any type of cell. They have a limited number of programs that they can follow to become, right? So they can become multiple type of cells, but not all, like if the totipotent, okay? Sarah, Amanda, or Jenna, could you just keep an eye on the chat? I really cannot follow 3 things at the same time.
Do blastocytes regenerate into any type of cell, like any type of cell?
Can you repeat that again, Elena?
blastocytes, can they regenerate into any type of the tissue in the organism?
Okay, so we go back into terminology.
Yeah.
The site termination is a mature cell, osteocyte, blastocyte. The site is the definite term. Okay, so that cell cannot go back to anything else. Okay, say osteocyte, fibrocyte, they cannot go back. The osteoblast term means that they can still regress. They are committed, but you can change their fate. It's still a little bit dependent on the surroundings. That explains a little bit more?
Yeah, it does. Then
okay.
What type of cells live in blastema?
So based on the paper, we say that the blastema are the specialized group of cells that has a little bit of stem cell traits for regeneration, but they need some signals, right? So now I have, say, signals, which should signal you that I'm going to ask about signals. So what are the signaling molecules responsible for regeneration in Planaria? Annika? Yeah, Annika, you have like a zillion annotations on that paper, so you were my girl to go for this question.
Well, the one that stood out the most to me, like I know they talked about, sorry, my dog's in here. I know the one that stood out to me the most was the Wnt. That's a
windlass, the WNT windlass.
Yeah. That's part of the beta-catenin pathway. Mm-hmm. So that one, so the more... Okay.
He's answering.
Yeah. So the more you have of the paracrine factor, you have more in the posterior end, and you have less in the anterior end. So that's your morphogen gradient. So then with the more Wnt or wingless, whatever it is, you have more beta-catenin activity, and then you have more of the activation of the TCF transcription factors, I think. Okay. So with that, the more you have, then you'll get the tail, and the less you have, you'll get a head. And I think like when you have amputation, you still have that relative like morphogen gradient. So by that relative gradient, they can like assess, I guess, what needs to grow back, if it's a head or a tail or.
Okay, so you talk about gradients, and you mentioned that the beta-catenin and windlass beta-catenin was more towards the posterior part, right? So what is in the anterior part? I'm asking you, and we're going to let somebody else to answer.
Okay.
Perfect, thank you. Not that I know that you know the answer, most likely because you have a question about that, but I just want to be able to hear anybody else. Unless, then you come back. Nobody? So what are the three axes that the planaria needs to regenerate when we cut it? And that was part of the lab report. So
longwise, it needs to elongate, and
so longwise is anterior posterior.
Yeah.
Dorsal
is one of the most like active portions that promote this regeneration. They need to regenerate the width, obviously, of not dorsal ventral, although in dorsal ventral direction as well, and to the sides, but I'm not sure
right,
yet.
Left.
Yeah.
Okay. So, Annalie, you say dorsoventral. What is the signaling pathway that determines the dorsoventral?
The BMP.
The BMP, yes. So Kaya and other people had it. Luis also had that one. So, and the lateral side, the lateral axis, what determines the lateral axis? Not necessarily, Luis. It does have a little bit of it, but it's not the beta-catenin wingless.
Is it the signaling pathway?
It's a signaling pathway. Well, it's a signaling molecules.
Hedgehog pathway?
Nope.
Nope. Okay.
No, hedgehog is implicated a little bit more onto the anterior posterior as a parallel, but not on this lateral side. Annika?
Is it the FGF for lateral medial?
It has a little bit of the fibroblast, but it's really a slit and windless 5. Remember that we have a lot of windless, right? And this is when you need to start paying attention to stuff, particularly with windless, because windless, we're going to mention the 2, we're going to mention the 5, we're going to mention the 7, we're going to mention the 3 a lot through the course. So we need to start distinguishing really which ones are which ones. So in this case, is the slit and windless 5, the ones that are from the lateral side. So we see that these molecules create a gradient, right, on those edges. What else do we have regarding this paper?
I know I didn't read over it fully. I read the other one, but I kind of skimmed over it, and I noticed something that that when these something happens in these pathways, the planaria can actually grow like a second head or a second tail instead of what it's supposed to, right? Yeah, I thought that was really interesting too.
Mm-hmm. Yeah, I found a video of that done in 2016 in India, and that Chimera planaria, understanding that heads don't really communicate with each other and they're just kind of trying to move where they want to, but they have only one tail, right? So they end up spinning around each other for most of the time.
We have done this in the lab, and we would have done it this year, where we split the head up to the pharynx in half, and we're normally gonna regenerate 2 parts of head, so it becomes a planaria with 2 heads. Okay, the things that we can't do this year, but anyways. So, yes, Maher.
Why did they call it the bone morphogenetic protein?
Why do we? It's a generic because it's normally implicated in regenerating a lot of the osteogenetic parts of the embryo. So cartilage and bone are regulated by the BMP. Okay.
Yeah, but we are talking about the planarian, right?
Yeah.
Planarian
because it's the same factor. Doesn't matter. That's what I say, that we use the planarian as a proxy because they also have it. Of course, a planaria doesn't have bones, but we see that fish, that is the next level, right? It plays the same role. So
it's the general terminology, I mean to say.
Yeah, but it's because from the molecular point of view, these are essential molecules and can be expressed in many different types of organisms. The action is more or less similar, but the end product is different depending on the group. Planarians don't have bones, but they have other tissues. Okay, So it's the toolkit that I said since the very first day that I wanted to give you. So Luis, yes, this is really interesting because there is a lot of investigation that is happening regarding how we can learn how to use these molecules to help human healing. We go through the process of regeneration for us when we have a cut, for instance, right? So, which brings me to one of this question. What is the main difference between healing and regeneration? It's mentioned in the paper.
Sorry?
It was mentioned in the paper. Go ahead, Maher.
Yeah, I think healing results in another type of tissue, like for example, if it's a scar, it's going to be a fibrous tissue. But regeneration, it's the same tissue, it's the same cell with the same function. So,
which is caused by, what did Emma say? And Ish? The blastema. Okay, so this is the big difference between both. Humans are capable of doing a lot of healing, but these are the FGFs factors, the fibro growth factors, because we're going to have a scar tissue. However, humans are capable of doing regeneration in some tissues, like the liver, right? We can regenerate the liver. We have grafts of liver. We can transplant liver, half here, and it will regenerate the entirety of what we have. Now, it all depends what type of part of that liver has been transplanted, right? Because we have alpha and beta cells in the liver, so we need to take the same part. Otherwise, those cells are predestined to become something, right? So, but humans, we do have some degree of healing, of regeneration, however, not as big as we can see in the second paper, okay? Okay, I'm just trying to, Amanda, are you keeping track of those comments? Okay, it's just, I just cannot go into different screens at the same time. Okay, so that paper of Planaria is really good just for that, right? That it shows how in a simple organism as the Planaria, we can have some same factors that we're gonna see as we go through, right? I know that some of you maybe in other courses, you cut a Planaria and yeah, I regenerated, but why? Well, this course, it shows you the why and how. Okay, so that's the most important. As we advance in our academic journey, we always need to build on the knowledge, right? So this is the level that I wanted just to bring. Sarah, you made a hand up. Just you wanted to comment on something, Sarah? My GTA, yes. It's just, I saw your hand going like this, so it's like, okay, are you? No? Okay. Does anybody want to talk a little bit more about this paper? Not all at the same time?
I guess I can comment on the BMPs, the sort of naming of it. It was I was interested in it too, so I looked into it, and it was like in the 1960s, I believe. It was first discovered in the bones of rabbits, and that's why it has that name. But nowadays, it plays roles in all sorts of development, right? It's kind of outlived that name.
Yeah, exactly. So as I say, it's just, even though if it's discovered in one, then you see that that same molecule appears in other organisms, accomplishing functions very similar. However, the output may vary because you can only do based on the genetics that you have. You cannot ask for some to behave in a different way. If your genetics don't have that, it's like, oh, yeah, whatever, right? I can't do the instructions that you are asking me because I don't have all the machinery to complete that task. Okay, perfect. I see. Mandy, thank you for replying to the question. So we're gonna split now to the next paper. the fish. And I know that maybe some people just go and say, oh, 16 pages, fuck it. No, I don't want to read that. It's too long. But I know that some of you read it because I downloaded all the comments. So come on, don't deceive me. Fish are my thing. Annika?
Maybe I can just comment on something. The thing I found the most interesting in that paper was that when they did the transplantation experiment with the tail, and that they moved it like it was either like medially to laterally or vice versa, the lateral part is longer and the medial part is shorter. But when they transplanted it, the medial tissue became longer than it would have been if it stayed where it was at first, but shorter than it would have been if it was a lateral tissue originally. So that was interesting to me to see that it was like taking a little bit of both. I don't know.
Yeah. And does anybody has an idea of why?
I think it could be because of the site, like where it's placed, which usually has an impact on the formation of certain body types, limbs, cells, etc.
Yeah, so we talk about that in lecture, right? That the environment determines to a certain degree what a group of cells will become, even though these cells are multipotent or pluripotent, right? They are receiving signals. And in here, we're talking about whether there is a top nearby the body or whether that part is a little farther away, because now we still have that gradient. One of the last chapters that we're going to be looking into this curve is the origin of the limbs. So, and that refers to some of the topics that this paper talks, the zygopod part, whether there is a proximal or distant part of the fin. And in this case, when you are, the caudal fins can, depending on the species, and here we are with the zebrafish, that fin has a different shape and length compared to the pectoral fins, right? So depending on where we put this, as I say a couple of minutes ago, you have some information already in your cells. Now we are inducing something else, so they're going to change their shape. It's not like, and somebody had a comment into this paper, so they're going to create and develop the structures with the same shape and size of the structures that were there before. Not necessarily, because if we look at when we amputate a tail, for instance, in an amphibian or a reptile, the tail that comes up is not necessarily the same length at the end. It's a little shorter sometimes. So even though it has all the instructions, those blastemas that regenerate that tail, the information is still missing a little bit. But it developed the right structures. We want to regenerate cartilage, bone, muscle, nervous tissue, and so on. But the gradient is not necessarily the same, right? Because remember, these are all signals that are based on diffusible molecules. And one of the problems when we depend on diffusible molecule is, what is one of the main problems when we rely on diffusion of molecules? And Alicia?
I have a question, but I don't really know how to phrase it completely properly. When I read the paper...
It's related to what we are talking?
Yeah. So...
Okay, go ahead.
When the caudal fin is regenerated, will it function to the same level as it was before it was, like when it was like first there, before it's regenerated? Like, does it...
It should be. It should be. In most of the cases, yes. Now, if the size is a little smaller, that may be an issue.
I also
read
in
theory.
it stated that it needed the wound, it like would regenerate as soon as the wound would close. So when the wound, if like, say the wound just never fully like sealed or closed, would the regeneration process happen if it kept being like reopened?
Yeah, but in theory, once it replaced the part, the whole process shuts down, because otherwise it would be a waste, right?
Okay, yeah, that makes sense.
Okay. Yes, diffusion limits by the surface area. So, and surface area in this case is distance to and size, right? So the longer those molecules need to go, the more diluted they become, right? So the bigger the animal is, the harder it will be for those molecules to pass the right message to the right structure, okay? And Luis, yes, you are right in that assumption that a lot of these molecules depend on these gap junctions to communicate. So a lot of these tissues may become impermeable to some molecules, but there are specific gap junctions that are open for them to be able to allow the communication. And that's the second theory that is out there. It's not just diffusion, because diffusion have these problems among distance, but if we use gap junction, then it will be different. So thank you for that point, Luis. Anything else regarding this paper?
Professor, in regards to cells of fishes or even planaria, do they have what is called the apoptosis or programmed cell death?
Yes.
They do have?
Yes, they do have.
Okay.
They do have. I'm gonna add a link regarding the planaria. If we have time today after the pre-lab, I'm gonna show that video. If not, I'm gonna put 2 links actually of the planaria in the D2L, and that explains a little bit more of the molecular basis by one of the planaria experts, that is, what is his name? Sanchez Alvarado. I can't remember his first name. He's one of the experts of planarians, and a lot of the papers that you read is based on that. And it explains really all this molecular cell communication regarding this. Okay. Anything else that anybody wants to add on this paper?
There's 2 things that I found that were pretty cool.
Okay, Annalie, go ahead.
That there was one thing that I found cool was that each fin recreates its own blastemas.
Yeah, they are fin specific too, right?
Yes. And there was one more thing. I found that the zebrafishes, the fact that they can regenerate organs like hearts, brains, spinal cords, and appendices, so fins, I found that pretty interesting too.
Yeah. Thank you for bringing that up. And that's one of the reasons why zebrafish is one of the best models to study. There is a lot at the Children's Hospital in Toronto. They have a huge system with zebrafish. It's in the Children's Hospital because the heart formation is the same thing in fish as it is in humans, even though they only have 2 cavities. 2 cavities, 4 chambers. We're going to see that when we see the heart formation in vivo. But the same toolkit happens in fish and in humans. So we can study a lot of the genetic conformations in hearts by looking at fish. So, and fish, for the nervous system, and particularly for the brain, had what we call the never-ending regeneration. So they are very, very plastic about it. If something happens, we can't regenerate that. One of my master's students, Mariam, she's looking at electric fish from Panama, and they have the tail, they have the electric organ that produces the discharges for communication. But they live in an environment where the catfish, Ramblia aquilans, inhabits too. And with the barbels of the catfish, they can detect the signal, the electric signal produced by this fish. And they just grab them by the tail. So a lot of the time we were like having fish that were missing half of the body, which corresponds basically to the part that regenerates these EODs, the electric organ dischargers. And in some of them, you could see the regenerated part. It's a little shorter, it's a little more blunt. So they lose a little bit of the secondary sexual characteristic that is in the tail, but they are able to regenerate the entire apparatus that generates the electric signals, which is pretty cool. And that's part of their ability of fish to have the neurogenesis. They have neurogenesis the entire life in fish. So this is pretty cool. So they have a whole whack of blastemas, of these multipotent cells in their system, in the nervous system, that is capable of regenerating all the time. That's what fish are so cool. Anything else?
Yeah, I had a quick question about the zebrafish article. I was reading the portion, and forgive me if someone's already asked this, but I was reading the portion where it says they can regenerate to their body's size, so they always stay within the proper ratios. What happens if a growing fish is amputated, so it loses something that may be increasing in size? How do they know? Does it grow back to a smaller size, to a larger size, or just kind of in the middle to grow with the fish?
It will normally go back to what it was before. But as I say, they can have some variance, and normally it's never bigger, but normally it's a little shorter of what it was.
Okay, so it can become smaller to grow into size, but it'll never really ever have a reason to regenerate larger than it should be.
No.
Okay, understood.
No,
thank you.
Those are more of environmental pressures that determine that an individual will become a little shorter or larger.
Okay, so but in general, it does always stay tuned to the body ratio, attempting to stay as close as possible. Okay, thank you.
Okay.
I also
Taya?
I also had a question.
Oh, Erin.
So something I found interesting with this article is it mentioned tumor suppressors' role in regeneration. And I did some research and I found an article saying that the P53 isoform, it could promote zebrafish heart regeneration. So that was pretty interesting.
Yes. There are so many things that we learn. And we're going to go next year or in 2 years, I'm going to go and see who cited this paper that I just put. And then it's going to be 20 more papers that started from this one. And every year we learn so much stuff. It's like, this is one of the courses that I absolutely need to update every time I teach it. Human anatomy and physiology, I can teach the same thing every year. And I just need to change my examples. Vertebrate physiology, I do the same. I almost teach every year, although I am updating every couple of years. But this course, every time I teach it, I need to update because we learn so much from the genetic point of view, from the cellular point of view, and from the molecular point of view. This course keeps me up tempo. I need to update every year. Things that I said a couple of years ago, I was speaking to you the other day, polyspermy at the beginning, 10 years ago, 15 years ago, I was like, oh no, just one sperm is needed to fertilize and no more. And if we have more than one sperm that goes in, and we saw the video the other day for birds, that's not true, right? If we get even 15, that's not enough to promote the embryo completeness, right? We need, how much they say, 100, more or less, a sperm to get into the egg so the embryo can develop properly. So this course can be, okay, Mary, get your gear in it. Okay, we're gonna stop right there. Thank you very much. You see, this is very informal. The more you participate in this discussion, the better we clarify questions and we can make more links to it, okay? Okie dokie, so having said that, I now need to, where is my, okay, here we go. So close the chat here, and now I'm gonna need to, oh, no, not yet. I need to go and put my pre-lab presentation. So today's lab is a little longer than normally it's going to be for the rest of the, oh, it's not this one, the one that I want to show. For whatever reason, I had like 25 open, windows open. This one, this one. As a full screen. There we are. Now we can have you guys in one screen and the presentation in the other one. Okay, come on. Okay, so just a couple of clarifications. So labs are due on Saturday by 10 a.m. Are we clear on that? Okay. I could have asked to be submitted tonight by midnight. However, I say Saturday at 10 a.m. Okay. And you have a full week to answer the questions. So it's more than fair. Okay. Because before we had the 2 labs, now you are all at one. So everybody needs to submit on the Saturday at 10 a.m. in the Dropbox. Okay. The first lab didn't have any pre-lab necessarily because that was to do the cuts and all of that, and we didn't do that. So I'm going to start with the lab 2, the introduction to Gametogenesis. To come back to this window where we are. Okay. So embryology versus developmental biology. This is something that I explained to you in the first lecture. Embryology studies the development of an embryo, allows you to construct and make an idea in our minds, the 3-dimension of the embryo. So this is what we mainly do in the lab portion of this course, okay? So we look at histological preparations in support with the book, and then you should be able to place yourself into what part of the embryo is developing. Developmental biology is the sort of the processes which organisms may use to grow and develop, and that's the portion that we do in lecture. Okay. My cursor goes to the other screen. Here we are. So normally we have a box of slides, and that has 40 different slides, okay? The way how we are doing it, if you already look into D2L, you have a PDF with the slides, photos, and some models that you would have access in the lab. So we're going to be providing the photos for each of the slides. Some of the photos will be at 4x, some will be at 10, some will be at 20, some will be at 40, and some will be with all immersion. Okay? So you are only allowed to use the photos that we are providing you. You are not to go and find a photo on the internet unless it is indicated. If we say label a photo, is the photo that we have provided you with on the PDF. Is that clear? Okay, perfect. So pay attention to the slide number. So if you look into the PDF that I gave, it says slide 1, slide 2, slide 3. And the models also have the models that is indicated in the document. Okay, so just for referral. Okay. So the name of the slides also, just in case I do a mistake, because we are humans and sometimes I forget to put something, just keep the name also in mind, that it coincides with the right photo. Okay. And then you are just to use the photos that we provide you unless otherwise indicated. Most of the photos that we took were using the compound microscope, but some on the later stages of development will be taken with the stereoscope. And you know everything about the lab manual, right? And how we're gonna do this. Okay, one of the first things that we need to take in consideration when looking at the slides are planes and orientations, okay? So planes can be sagittal, okay? Could be also transverse. So sagittal, we have an anterior, lateral, left and right. We have transverse when we have anterior, posterior part, okay? And we can also have the frontal. Well, this is the frontal, okay? We're gonna have these cuts in here. Then orientations of the view, okay? So we're gonna have cranial or anterior. We have dorsal, we have ventral, and we have caudal or posterior, okay? So when you are looking at the slides, whether there is a cross section, okay, or a transverse section, you gotta think first about that cut, because that would allow you to say, okay, so if this is a transverse cut, I'm supposed to be looking at an anterior or a posterior portion of the embryo, okay? So that allows you to mentally plan what you should find at that specific section. Did I make myself clear on that one? Okay. So as I know that some of you already took histology, but every year we had this right Monday where we say, oh, why I don't see that? Well, because you are in the anterior part of the embryo and the structure that you need to look is in the posterior part of the embryo, especially when we have a serial cut, okay? So pay attention to that. So this is very important when looking at histological preparations. So which brings me to the next point, the slide orientation. So later in development, we want to have whole mount or in situ. So we're going to have this taken with the stereoscope. These are big embryos. Then we have a section that has no orientation. Simply, it was cut, like here in this blastula, okay? Then we're going to have a cross section or transverse section, either going to be a CS or a XX, okay? And this is in the anterior portion of the embryo. These are the eyes, this is the crystalline, and this is the telencephalon. And this is the neural tube, okay? And this one here is the notochord. So that's how I know, oh, it's in the anterior because I'm looking at these structures, okay? Then we have the sagittal section. So this is the anterior portion, and this is the posterior portion. These are the somites, and this is the diencephalon. Okay, so knowing which cut you have will allow you to place yourself a mental map. Oh, okay, I should find these structures here, okay? In the serial cuts can be transverse or longitudinal, okay? So also pay attention to it. So in this case, we have this one. This is A, B, C, D, and this is this way, okay? So you're gonna see that in your book, it says, look at section F. So if you're looking at section F, this is more or less where you are placing yourself, okay? So you know that in this section, okay, maybe you're gonna find the hindgut, right? Maybe you're gonna find the limb fields. This is gonna be one of the legs, and this is in E. This is the wing field. This is the wings. Okay. So depending on the position in this serial cut, you're gonna find different structures. So once again, pay attention to that. In the lab notes, you really need to pay attention to the level at which the cut or the structure is made. Okay. Then this is an example to show that a cross-cut at the anterior level of the embryo is showing different tissues that when taken at the lower part. So here still is the anterior portion of the embryo, right? This is the neuropore, the cranial neuropore, okay? And this is the cross-section of this embryo, okay? So we have here the neural fold, the neural groove, the notochord underneath. We have the endoderm here, and we have the lateral mesoderm. If we do it a little more towards the back, then we see different structures and how it evolves. So pay attention all the time at what level we are having this cut. So the hair, if we have a little butt, the hair may appear on this section here. It's not there, it should be around here. Okay, so as explained it yesterday, in this course, as you have seen already, we will be looking at the development of different groups of animals. So we're gonna be using some protostome as a base, as the sea urchin. Some examples that we're gonna see, very few, but we're gonna mainly focus into the deuterostomes. So protostomes in Greek means first mouth, okay? That's from Greek. So I include all the animals with a true coelom. They all have a spiral segmentation, cleavage, and the blastopores becomes the mouth. And we explained it yesterday. The deuterostomes are also from the Greek, that is mouth second, okay? And includes all the animals with a true coelom, radiant cleavage in most of them, okay? And the blastopore will form the anus. So we're gonna be looking at sea urchin, starfish, fish, frog, chicken, and humans. And as we saw yesterday, we started with the sea urchin, and then we are now on fish and frogs, okay? So the sea urchin is one of those that we really use in the lab because it has a lot of eggs when it produces. So it's very traditional organism for developmental biology. Embryos develop very, very quickly. They are transparent, and we can identify the structures very well. The starfish is also used very commonly in developmental biology. This lab has one component on it that you're gonna be looking at the polar bodies on the starfish. The other sides of embryo are transparent. They're small though, but you can use them a lot. Whitefish, we're gonna be using to look at the stages of mitosis. If you took anatomy and physiology, for instance, when we show you the slide with the different mitosis stages, all those are made with the whitefish because it's easy. Then we have frog, standard model organisms for developmental biology. The embryos are very large and very robust, so we can subject them to different treatments and see their development. Then we have the chicken, classical model, also because it has a development that is very, very, very similar to humans. And since working with humans is a little bit more of controversy, so we can use the chicken for that. And in this lab, you gotta break an egg and find all the different parts as indicated in the book. And then mammals, but of course, we cannot use humans, and so we have different embryos that we can work in other animals, use as a model, but not for humans necessarily. So when we come to this lab, we're going to be looking at these stages of development, stopping here. We don't see the larval stage or maturity stage, but we want to see all of these up to organogenesis. So today we are in gametogenesis, and next are the other ones. So gametogenesis is simply the production of gametes through meiosis, as we saw in class. Chromosomes goes from 2N to 1N haploids. The spermatogenesis is the production of the male gamete, the sperm, while oogenesis is the production of the female gamete outside ovum. Spermatogenesis goes from primary spermatocyte, mainly in the outer layer of the testicles. These cells are 2N diploid. They go under the first meiotic division, and then we have split the homologous parent chromosomes. And then we have two cells that form, but these ones then are 1N, and that is at the end of the first meiotic division. And at this level, there are secondary spermatocytes. Then this one gonna go to the second meiotic division, and each of the secondary spermatocyte produces, of course, two more cells. So we find out that we have at the end 4 spermatids here, and they all go to the process of spermiogenesis. So all of these cells normally have a cytoplasmatic bridge in between them, and spermiogenesis is the process by which these cells become individual sperm cells. Spermiogenesis is not part of this meiotic process, but it's part of the maturation process of these sperm cells. So I use this image to go a little slower. So we start having the production of a sperm happens in this seminiferous tubule. So you're going to have a photo. You have a photo, I think it's at 4, 10, and 20, a little bit more, I think, up to 40 of these seminiferous tubules, okay? So here is this over here. So this is where the sperm are produced, okay? And during this process of maturation, cells are going to start going from a spermatogonium that is going to be found more at the edge. Let me just grab here, more at the edge of the cell over here, that is spermatogonium. And then primary and secondary spermatocytes, you're going to see a little bit of changes in the nucleus. The nucleus start becoming a little more condensed, and cells become a little smaller as they mature, okay? And at the very end, the spermatids are really at the lumen section of the seminiferous tubule. So we're going to have them a little bit more on this end. So through the photos that I provided you with, you're going to need to identify the different type of cells based on the images that you have in the atlas and the book, okay? So at the end, most of the cytoplasm of the sperm has been eliminated. The nucleus is haploid, and we have the mitochondria packed. So this is the same image that we saw in lecture, where we see this is the sperm, just to the point, our immature cell. We have the nucleus, we have the Golgi apparatus that is condensed to become the acrosomal vesicle. That's what we saw in lecture. The centriole will give origin to the flagellum. Okay, so and here we see how this acrosomal vesicle, as it starts changing to a secondary spermatocyte, in here it becomes really all around the nucleus, and the flagellum is a little bigger. Mitochondria start migrating into the mid-piece. Here we see them all aligned, okay, in the spermatocyte cell. You still have a little bit of cytoplasm. And remember, these are, oh, come on. There we go. This is the bridge that connects one spermatide with the other one, okay? Oops. And at the end, all the rest of the cytoplasm is reabsorbed. And at the end, we have a head that contains the nucleus, a mid-piece that contains the mitochondria, that is the powerhouse to produce ATP. So the tail or flagellum can whip and swim. And then we have the end tail, okay? So remember that I told you that males rely on the Raffle ticket production of hundreds of millions of spermatocytes, but only a few are capable of, I'm gonna look at the chat in 2 minutes, okay? Only a few gonna find themselves nearby the egg. I say that normally males gonna produce something around 250 million sperm, well, not produce, but ejaculate 250 million, and this is in humans, of course, spermatocytes. And only 200 gonna get close to the ovum. But a lot of those are incapable to arrive there because they present a lot of abnormalities. So we're gonna have here this a bent tail. So this is the head and the tail is completely bent. We have some that has absolutely no tail. We're gonna have some that have curled tail. Try to whip with a tail that is curved like this. It doesn't gonna be easy. Some gonna have some droplets. Just like if you have something in the middle that is impeding them too. Others gonna have a shape of the head that is not proper, and some gonna have two heads, for instance, and some you're gonna have two that have two tails too. So this is all in bull sperm, of course, but a lot of the sperm that a male ejaculates have a lot of abnormalities, and that's one of the main reasons why they never get where they need to get. So it's gonna affect the motility of that sperm. You have also some, two images, I believe, with different type of sperm. So you have the frog sperm like this. You have the rat that is like a hook. You see it here. The ball is very similar to males, humans. The rooster is a little more like a sigmoid type of thing, different than the rat, because the rat is really like a hook, okay? And if we compare the size of this is the starfish egg, and that tiny little thing over there in green is the male sperm. So proportion is really dramatic in this case. When we go to oogenesis, this is the same image that I show in lecture. So we have two things. We have the follicle, and then we have the oocyte, okay? So we have the follicle section, and before birth, we get into primordial stage, primordial oocyte in the primordial follicle. You're gonna see that in the slides is surrounded by more of a squamous, one layer of squamous cells, okay? Childhood, so at each month, this primary oocyte gonna start changing, okay? So after that, we're gonna have one layer, the primary follicle at this stage gonna be not as squamous, but more cubic cells around one layer, okay? Then it becomes into 2 to 3 layers. That is the growing follicle, and that is still arrested in prophase 1, okay? Then, because of the growing of the follicular cells surrounding the oocyte, we're gonna have here a little, what we call the antrum, excuse me. So from primary oocyte here, that we have 2, 3, up to 5 layers of these growing follicle cells, to the secondary oocyte that we have in here. Here we're gonna see the antrum. So this little blue space here That's the antrum. And that is when we know that the cell is growing, okay, to become mature. A lot of books gonna have this as a secondary follicle. You have a couple of photos where you have this small antrum, and you have some where the antrum is bigger. And that is really, when that antrum is really big, that's the tertiary or Graafian follicle, okay? And then, so at this stage, the egg is arrested in metaphase 2, and gonna only continue the process of meiosis if fertilized, okay? And here, then we have the ovulated secondary oocyte, and these follicular cells become, excuse me, the final, and now the name comes in French and it doesn't come in English, le corps jaune, the atrophied follicular cells.
Corpus luteum, is that it?
Yes, the corpus luteum, and then it's the corpus albicans. Thank you, Catherine. So you have a photo of that corpus luteum in here, and then the photo is really nice because it's yellow. Okay, so you're gonna have that one, and then once they're generated, it becomes albicans because it becomes white. Okay, so when it goes to metaphase 2, we need to remember this is 2N. Up to here is 2N, right? It's diploid. But in order to be fertilized, it needs to be 1N, right? So once before it's released, we have our first polar body. And if it's fertilized, it continues and then it needs to release. Here's a second polar body and can be fertilized completely. So at the end, we have 3 polar bodies produced, and this one is just N. And this one just is mainly cytoplasm, okay? So in here, we have the different stages through which this egg is going. And we have a Graafian follicle, big antrum, right? Here we have a secondary because it has a small antrum, okay? Here, this is one of the primary because it only has 1 layer of cells. And this is more like the secondary because it has more than 1 layer of cubic cells all around. So when we look at a human egg, this is one of them, okay? We have the secondary oocyte, we have the zona pellucida all around, and we have 1 polar body. Is this oocyte fertilized? That is a question.
So did you just ask if
this oocyte is fertilized?
No.
Yeah.
Who says yes? Who says no? My eyes wasn't as quick to switch to the screen to see who was talking.
Helen says no, because the polar body is still in the cell. I think that this oocyte in the process of developing because the polar body is still there. And
okay, Elena, how many polar bodies are there?
1.
1. So it is non-fertilized because it's only had 1 polar body. Okay, that's one of the techniques that is used in vitro fertilization to know if the egg was fertilized. It's not because we went through division, because as I mentioned before, the segmentation in human takes long time to occur, right? But if it exposes the second polar body, then you know that it's fertilized. So that's a better signal for in vitro fertilization, okay? So here is the chicken egg that you are going to be doing at home, and you can eat it after if you eat eggs. If you are vegan, well, you may not have eggs, but so follow the instructions that are in the book, in the notes. So you need to break the shell very delicately. I normally ask you to open that egg at the beginning from the bottom so you can see the membrane, the air chamber that is located at the larger part of the egg, not on top, but here, okay? And the wider portion of the egg. And then you can open very delicately so you can see the different things. So you're gonna have to identify the shell. Then if you open and put it into a surface that allows you contrast, that's what I say, put it on a pan, you're gonna see that there is a thin albumin, then it's a thick albumin, okay? And then you're gonna be the chalazo, either strand, and then you're gonna have the yolk with the vitelline membrane that is here. And one of the points is that the germinal disc, doesn't matter how you open the egg, always the germinal disc or blastodisc is on top. So you wanna be just a very tiny whitish point at top, okay? And here we have the inner shell membrane, and then we have the outer shell membrane. So when you break the egg delicately, you're gonna see those two layers of membranes there, and then you have your air chamber, okay? And that's it. Oh, didn't change my, there we are. My pointer is saying, it's been an hour. That's my writing. No, the labs cannot be done in partner or individually. Okay. Yes, Elena, evolution in a single cell.
Not any other cell that undergoes such drastic changes?
Can you repeat that question again, Elena?
Is there any other cell that undergoes such drastic changes while still being the same cell?
Mm-hmm.
I'm not talking about
yes.
Stem cells going into any other type of cell, but like the way germ cells, sperm cells in particular, grow.
Well, it all depends on the cells. Not all the cells can become anything or something, I should say. It all depends on the type of cells. But the egg is one of that totipotent, because from that cell called the oocyte, we're going to generate all different types of cells of an individual, right? Did I answer your question?
More or less.
Okay. So the notes for this lab are in the D2L. Now, these labs are not like a formal lab where you have an introduction and you have materials and methods, and then you have a discussion. Just limited to follow the same format as the lab notes, okay? Put the titles of titles, use the same numbering as in the lab notes. So for us to be, okay, this is section 1, this is section 2, this is section 3, this is question 1 or 2 or 3 for part 1 or 2, depending A, B, C, D, whatever it is, okay? So no introduction, no materials and methods. Remember, this lab was just you guys going to the lab, finding yourself the slides and finding everything, and I needed to transform this into a lab. So virtual lab. So this is the best that I can do, is individually work. If you you have many questions that need to be answered fully. The more detail you put, the better is for us to be able to mark. Reference need to be citations for where you got that information. Okay, if it was Wikipedia definition, it was Wikipedia definition. Okay, but citations, that's something that I insist a lot for all of my lab citations. You need to grant the benefit of who did that. For this week lab, we need to. Mandeep, I don't understand what your question is. The notes are on D2L. The photos are on D2L, and you have your book. The lab book is the one that you need to use to read. We say read each section in the book and answer the following questions. And I just need to do one thing in here. Just give me a second. Trying to go to the lab content.
Is it the discussion about the lab 2 already?
Sorry? I'm just trying to catch up. I was uncovering the manual.
Is this the discussion for the lab 2 report?
Yeah.
Oh, okay.
Yeah, so you have questions. In each section, you have questions. Read gametogenesis and answer the following questions, okay? And provide the citation. If it's the lab atlas, cite the lab atlas if you obtained your information from it, okay? It says, look at figure 4.3 in their atlas and label the following. Label this slide 1 based on that. So use that book, take the photo from the PDF, which is not locked. You can grab that and put it into your report, okay, and label it. And you need to create a caption for that figure and so on. Yes, Sarah, thank you for replying. You have an idea, it's not your own, just put it. Derivation is the origin. So I'm gonna say is Greek, is Roman, is Latin, and it means this. So zygote, zyg means something. Mesoderm, meso means X, and it comes from Greek. That's the derivation.
It seems like
tomorrow morning is due the Planaria Lab. Tomorrow is just the Planaria. Okay. Next Saturday is lab gametogenesis. In the syllabus, you have due dates. I haven't changed any of those due dates. Most of the information is all in the syllabus. So Raven, no, Antrum gametogenesis is due next Saturday at 10. Yes, all the labs are due on the Saturday at 10, but for the following week. Okay, so tomorrow is Planaria. Next, that's the 5th, I think. I can't remember which day it is. Next Saturday is this lab 2. Now, do you have any questions regarding the Planaria report that is due tomorrow at 10?
Yes, Professor, in regard about the final day, do you refer to the pictures that were sent on the on D2L? Because they seem to be unclear a little bit.
Can you ask your question again? It was a little gap when you were speaking.
Yeah, in lab 1, the Planarian, the questions that came after the final day part, where it said the final day.
It's based on the photos that I gave you.
Yeah, the photos, yeah, but they were very unclear. So, like when they
just do the best you can. Okay. I couldn't get Planarias because we couldn't order anything. So I just tried to grab some photos from previous years. And some students left some photos in my laptop, in my laptop. But most of the students, I asked them not to leave any photos in the laptop, but take them. So I didn't have that much. So just label as best as you can.
I have a question about the next lab, the Gametogenesis one. For chapter 4, the Oogenesis, it says that we have to look onto page 47, 49 for the figures. So I'm assuming that's for the atlas. What happens if our atlas hasn't delivered yet?
If you look into D2L,
yeah,
there's a link for the PDF.
Okay. Oh, okay. Thank you.
Am I the only one who
There is no syllabus for the lab. It's one syllabus for the entire course, and it's in the lecture portion, not in the lab portion. My syllabus doesn't have this is the lab and this is the lecture. It's all one single document.
In my Planaria Lab, for the pictures, I have 2 sets of photos for Planaria Regeneration of the head, but not for the tail. Is this how it's supposed to be, or something wrong?
Both, because you have both sections. You have the anterior and the posterior portion. You need to discuss both.
So there are some pictures of the tail regeneration.
Yep.
Okay.
There are 2 photos per day, anterior and posterior, 2 pieces.
Ah, okay.
Yeah, so I just need to, once I'm done with this, I think you need to, for whatever reason, Joe, for you, you didn't need to ask me permission, but the students need permission. This is odd.
Permission for what?
To open the, remember that I sent you the link just to see?
Yeah.
The students need to ask permission, and this is what I wanted to avoid, to receive too many emails.
I was able to just click on it.
I will do it.
Okay.
Yeah, and I did the same with Mariam, and she was able to. So I will give you permission as it comes. Okay.
Professor, I am not able to access the lab 2 on my D12. It's asking me to get access first.
Yes, Maher, that's exactly what I'm saying.
Okay.
That I will give you permission as it comes, okay?
Okay.
So if you all send me, I don't want to do this right now here with you, but I will do that. And so all of you should have permission once I'm finished giving it. Any questions?
Yeah, I have a quick one. For the lab, is it okay if it's just in a question and answer format, or would you like it set up in more of a paragraph kind of way, like set up to give you a structured, something more structured, I guess?
Well, the answers is not a yes or no, so you're gonna develop a little bit, right?
Yes, that's completely fair, I guess. Okay, thank you.
Kaya, you have a question? I see your little hand up. Kaya?
No, sorry, I already asked it.
Okay. No, as I say, no intro, materials, and methods. Just answer the questions, label the figures as asked.
I have a question about the video. So the outline is due tonight, right? There was
the outline is due this afternoon at 4.
Yeah, I didn't find any Dropbox for it, so I sent you an email.
It's in the same box as the video thing.
Oh, we're supposed to submit it into the video?
Yeah, I say, okay. and I put a note in the news. I say the video, the abstract, and the outline. All 3 things goes into the same folder. And as I say, only 1 person per team needs to submit. I don't want to receive 60 when 2 are the same. So all the 3 things need to be submitted into the same folder.
Excuse me, Professor, regarding the 500 words report about the defect, we have to both, me and my teammate, to provide 2 or just only 1? As
I just say, only 1.
No, I'm not talking about the outline. I'm talking about the report at the end with the video.
The abstract, just 1, because both of you need to work it out.
Okay.
Just 1 per team.
So a total of 3 files, 1 for the video, the other for the abstract, and 1 for the outline.
Yeah, exactly.
Okay. Thank you.
Yeah, I don't receive that by email because you don't know how many emails I receive per day. So if you send that, I may just open and forget to download it, and that's it. So it's better to have the all centralized in the Dropbox.
Yeah, sorry, my attention is so bad lately that I might switch back and forth and pay attention at the beginning of the sentence, but have no idea what's going on in a few minutes. And apparently I'm not the only one.
The 500 words is a maximum. Jenna, as I have said, I think this is the 7th time. The outline is to make you guys to work together, just 1 page, give me the big lines that you are have found so far and that you think you are going to include in your video. As I said before, this may change with time as you continue your research, but it's just to show me that you already started the search, that you have been working together into building this video. Okay, so 1 page is more than enough. As far as I get an idea of what are the topics that you can, so I can guide you and say, well, I think this is going too far away. I think you need to add a little bit more of that. Have you thought about looking into this? Okay, but I need to have something to better guide you. So 1 page is enough. Most of it is just a point form, right?
I have a question about the lab reports. So I wrote my lab report in a format that the questions are written in Times New Roman 12 and I'm answering in Arial 12. So they look different and the size is different. Is this acceptable? Is this not acceptable?
What type of character you use, I don't care. We don't care. As far as you see the question and right below the answer, that is what is most. You have questions.
Oh, the answer?
On top. Question on top, answer below.
Okay.
As far as it's clear that this is the question and this is the answer, I don't care, because sometimes the students try to answer all of the questions like in 1 paragraph, and then we don't know to what question specifically the student is answering. So I always say, put the question, write the answer. Put the question, write the answer. It's easier for the TAs and all of us. It makes our lives easier to do the marking. Okay, if you don't have any more questions, I am late for a meeting too. So I just look at my email on the other end, trying to accept all your asking permissions.
Good luck.
Yeah. So if by tomorrow morning you guys don't have access, send me an email, okay? And I will try to give you access.
Lab objectives. This activity allows students to investigate how planarians regenerate.
This activity allows the students to understand the highly coordinated aspects of gene transcription involved in regeneration. Planarian Background. The planarian is classified in the class Turbellaria, which is in the phylum Platyhelminthes. The freshwater tubularians are found in almost all aquatic habitats. Planarians hide under rocks, leaves, and debris to avoid light. Planarians have no definite. The planarian is classified in the class Turbellaria, which is in the phylum Platyhelminthes. The freshwater tubularians are found in almost all aquatic habitats. Planarians hide under rocks, leaves, and debris to avoid light. Planarians have no definite eyes, just eye spots that cannot form an image and are sensitive to light. Their information is embedded in the cerebral ganglia located just below. Planarians have a pharynx with double function both for the ingestion and evacuation of food. Planarians are known as trichyds because of their triple gut with a single anterior and two posterior branches. Planarians reproduce both sexually and asexually. In sexual reproduction they produce summer eggs, which are thin-shelled and transparent, and winter eggs, which are usually black and set on stalks. Planarians are hermaphroditic, possessing complete male and female systems. They produce asexually by fragmentation and tail dropping, in which they spontaneously drop their tails and each end regenerates the missing part. Planarians are carnivorous, eating both living and dead organic matter. Kept in captivity, they feed on raw liver. Lab materials. The photos you are receiving. This activity allows the students to understand the highly coordinated aspects of gene transcription involved in regeneration. Planarian Background. The planarian is classified in the class Turbellaria, which is in the phylum Platyhelminthes. The freshwater tubularians are found in almost all aquatic habitats. Planarians hide under rocks, leaves, and debris to avoid light. Planarians have no definite eyes, just eye spots that cannot form an image and are sensitive to light. Their information is embedded in the cerebral ganglia. ganglia located just below. Planarians have a pharynx with double function both for the ingestion and evacuation of food. Planarians are known as trichyds because of their triple gut with a single anterior and two posterior branches. Planarians reproduce both sexually and asexually. In sexual reproduction they produce summer eggs, which are thin-shelled and transparent, and winter eggs, which are usually black and set on stalks. Planarians are hermaphroditic, possessing complete male and female systems. They produce asexually by fragmentation and tail drop. Dropping in which they spontaneously drop their tails and each end regenerates the missing part. Planarians are carnivorous, eating both living and dead organic matter. Kept in captivity, they feed on raw liver. Lab materials. The photos you are receiving today were collected in our lab by students. We made the cuts as observed in Figure 1. Using the photos provided here, please complete this lab report by answering all questions and labeling the figures provided as indicated. Figure 1. Cut of Planaria. The regeneration of the Planarian was followed for 2 weeks. Please pay attention to the questions asked and be sure to answer them fully. Please provide a citation for all of your answers. There is no such a thing as common knowledge, information always comes from some reported studies/article papers. You should also have a citation section at the end of your report. The Planarian regeneration is normally assessed by monitoring how long it takes for the photoreceptors to appear. The photoreceptors appear as black dots located inside the pale round eye. In the example of a regenerating Planarian shown below, Figure 2, the eyes appear around day 4 and the photoreceptors on day 6. Figure 2. Regeneration of Planarian through time. Questions to answer for the report. Day 0 to day 7. What environmental variables do you think may influence regeneration? you.